this as well.
See how loudly you can speak.
All good. So hello everyone. So my name's Aviya Naccarella and I'm a PhD student at Deakin University.
And I'm Emily. I'm at University of Melbourne studying a PhD.
And so we're actually going to do a bit of a joint presentation to begin with, and this component's looking at mammals, fungi and plants, a tripartite relationship for a healthy ecosystem. And just before we begin, we'd just like to acknowledge the traditional owners on the lands on which we are all gathered here today, the Tauntarong people, as well as acknowledge the traditional custodians of the lands on which we conduct our research right across Victoria, and pay our respects to elders past, present and emerging.
Thanks. So because our PhD projects are somewhat of a similar background, we thought we'd do a joint kind of presentation to start off with, just looking at this three-way relationship between plants, mammals and fungi. Then I'll go into a bit of detail about my PhD project, which will be looking at the interactions between Potoroos, fungi and fire. And then Aviya will follow up with her PhD project, which is looking at the role of mycophagous mammals in ecosystem restoration. So we've already heard a bit about mycorrhizal fungi today from some of our other speakers, so this might be a bit of a review slide for all of you. So these mycorrhizal fungi are able to form relationships with plant roots, where they help provide the plant with important nutrients such as nitrogen and phosphorus, and also water. And in return, the plant provides them with carbohydrates. And these associations are incredibly important. Around 90% of all vascular land plants form mycorrhizal associations with fungi. Fungi have other kinds of effects on plants as well. So they can also confer increased resistance to different kinds of pathogens for the plant. And they also have indirect effects on plants through the action of their hyphae in helping structure soil. So these hyphae can spread out through the soil and help increase aeration in the soil, which leads to more water getting to go into the soil. So they have all these other effects on the ecosystem as well. The fungi that we'll mostly be looking at today are the ectomycorrhizal fungi. So just a reminder, these kinds of fungi aggregate their hyphae into a kind of sheath, which covers the tip of root tip cells. So just like these little pictures down the bottom here, that white fluffy stuff is the fungal sheath. And it's this interaction here at this interface between the fungal hyphae and the root tip cells, where this exchange of nutrients and carbon takes place between the plant and the fungus. Next one, thanks. So mostly when we talk about fungi, we're often looking at these beautiful big above ground fruiting fungi, for example our mushrooms. But we have an incredible diversity of these underground truffle-like fungi as well. You're probably familiar with the true truffle, which is the expensive truffle that you buy and you grate over your pasta. But we have all these other different kinds of underground truffle-like fungi. They've got all these different kinds of fruiting bodies, so they can come in all different kinds of colours. In general, they often all have a similar kind of structure. So they have an outer layer called a peridium, and this encloses a spore mass on the inside, so it protects the spores on the inside. Here in Australia, we've got an estimated 1,500 species of truffle-like fungi, which is a really huge diversity, and it accounts for 1/3 of the world's total. Most of these species are also endemic to Australia, and most of them form ectomycorrhizal relationships with trees. Some of our other speakers have briefly touched on this as well, but it's thought that the reason that we have such a high diversity of ectomycorrhizal fungi here in Australia is because a lot of our soils are quite poor in nutrients, so this really helps promote this kind of association where fungi are helping plants to access adequate nutrition and to really thrive in the landscape. These truffle-like fungi have evolved multiple times in Australia across multiple different families. So this really indicates to us that there's something quite advantageous about being able to produce a sporine body underground. So the most obvious reason is that it gives them a lot of protection against the elements going on in the weather and above ground. In the typical mushroom season, you have a lot of rain and a drop in temperature, and then our above ground fungi, such as our mushrooms, start to produce their sporing body. But if there's a change in temperature or the climate dries out and you have a couple of days of dry weather, these fungi can start to dry out themselves before their spores might have properly matured, so they aren't able to have viable spores dispersed. But if you're producing a below ground fruiting body like our truffle-like fungi, these can be nice and protected and insulated in the soil, so they're not as prone to drying out. And it's thought that some of our big aridification events in Australia's climatic history have been playing a role in the evolution of these protected underground truffle-like fungi. But there's had to be another kind of evolution that has occurred along with these truffle-like fungi in order to enable them to move their spores around.
So spore dispersal is really important for fungi to extend their range or colonise new habitats or even for genetic mixing. And so if we think of our typical above ground fungi, such as our mushrooms, their spores can be really easily dispersed by wind or water because they're exposed to the elements outside. But if we compare this to our truffle-like fungi, so those that fruit underground, they often need to be actually unearthed for their spores to be dispersed. And in many cases, this is where animals come in. So it could be reptiles, birds, or invertebrates, but in many cases in Australia, our mammals actually play a really important role in truffle-like spore dispersal. So in terms of our the diversity of animals that we have in Australia that have been found to feed on fungi, we've got 59 different mammal species, 13 bird species, 9 reptile species, and an unknown but likely very diverse range of invertebrate species. And so you might be quite familiar with some of these mammal species, such as the swamp wallaby. They're really widely distributed across eastern Australia, and they might even be, in your paddocks at home, for example. We've got a lot more cryptic small mammal species such as our dunnarts or our woylies or even our bandicoots and bettongs that you might be familiar with. In terms of our birds, the superb lyrebirds have been found to feed on truffle-like fungi. And really interestingly, there's actually been observations that suggest that there's an association between the eastern yellow robin and the superb lyrebird. So unlike the superb lyrebird, the eastern yellow robins are quite small birds. They've got very petite legs and they can't actually dig at the soil and unearth the truffles themselves. So instead, they figured out that they can go back in after the superb lyrebird's finished foraging in an area and actually scavenge around and eat some of those truffles that the superb lyrebird might have missed. And then, as Alan mentioned, we've got our eastern blue tongues, for example, that feed on fungi. And then in terms of invertebrates, species like the fungus-fly, for example. And so when animals disperse fungi, they can do so in a number of different ways. So the most obvious way is via direct consumption. So our long-nosed Potoroo here, for example, feeds on fungi and in doing so disperses the spores in their scats, so their poo, their faeces. As Emily mentioned previously in an answer to one of the questions, the spores actually do remain viable and undigested when they pass through the gut of the animal. They can also disperse spores incidentally via digging for other food sources, such as the eastern barred bandicoots, for example. Potentially when they're digging up soil, looking for invertebrates like worms or beetles, for example, they might either disrupt a spore body or they might just move soil which has spores within it. And then a really important but largely overlooked mode of dispersal is actually secondary dispersal. So this is where we have some of our predator species, such as our masked owls, our dingoes, or even our wedge-tailed eagles, when they actually prey upon some of these smaller mammals like our Potoroos or maybe our bush rats or swamp wallabies, they actually then disperse the fungi that these small mammals were feeding on in the first place. And this is thought to have really important ecological roles for long-distance dispersal. So, for example, the dingoes have been found to disperse fungal spores up to 10 kilometres away. And when we compare this to the dispersal capacity of some of our smaller mammals, like our swamp wallabies, they can really only disperse spores up to around 1 kilometre away from where they consumed that fungi. So when we think of that in an ecological sense, these dingoes are playing a really important role in dispersing fungi potentially across more fragmented landscapes, between different types of vegetation, or even between different ecotones, for example. And then another really interesting example is our dung beetles. So dung beetles obviously help to bury feces, and this helps the spores come into direct contact with the soil and plant roots as well. And really interestingly, there's a specific species of dung beetle that's actually been found to have an association with small mycophagous mammals, such as our bettongs, for example. And they actually live on the backside, so the fur around the backside of the bettongs, and then they're ready to converge on the poo as it's excreted by the bettong, and then they can fall to the ground with the faeces and go about burying the faeces nice and quickly, so they don't have to wander around the forest looking for faeces.
Thanks. So we do have a really high diversity of mammals in Australia that consume fungi. And they have different modes of consumption of fungi. Some of them are quite generalist, so they'll munch on a fungi if they happen to come across one, whereas others are really specialised. Our long-footed Potoroo, for example, consumes its diet of fungi as up to 80% of its diet, so it's super specialised. The long-footed Potoroo belongs to the Potoroidae family, which includes some of these members on the right-hand side of the slide, namely the Potoroos and the bettongs. And they are super specialised and typically consume a lot of fungi in their diet across the year. And often they're consuming a really high abundance of these underground truffle-like fungi. We do have other animals in Australia that Aviya's just mentioned that do consume fungi that are more generalist. So these include animals like the swamp wallaby or the bush rat. So why are all these animals eating mushrooms? So originally researchers weren't that sure that fungi could provide a really great deal of nutrition to mammals, probably because of their really high water content. But on the other hand, it actually could be that this high water content of these underground truffle-like taxa are actually helping mammals to survive in environments that are quite scarce of water. And some people have suggested the idea that these underground truffle-like fungi are playing a role in helping there be such a high diversity of small mammals in dry areas like dry woodlands in Australia. More research more recently has shown though that fungi have a really great variety of nutrients and energy that can differ quite a bit between fungal species. But the other thing that we have to think about is the particular mammal that is consuming the fungi. So our fungal specialists, like our Potoroos and bettongs, they have these really specialised structures in their digestive system that allow them to really break down fungi and get out as much nutrients and energy as they can. Whereas the more generalist mammals, they don't have these specialised digestive structures, so they might not be able to really thrive on a diet that's high in fungi, which may be why they only consume a little bit of fungi to supplement their diet. So while these mycophagous mammals are relying on fungi as a food source, whether it's only for part of the year or for all of the year, these fungi are really reliant on mammals and other animals for their spore dispersal. So they need a way to attract mammals towards them, as Alan has just discussed. So, for example, there have been experiments that have found that eastern bettongs are really attracted to the smell of mesophilia. So we know categorically that they are producing compounds that are attracting mammals.
So within Victoria, there's been 24 different mammal species that have been identified to feed on fungi. And some of them are our fungal specialists that Emily just mentioned, so our Potoroos. And then we've got a huge range of generalists that obviously use fungi to supplement their diet. And you might be familiar with some of them, such as the bush rats and the swamp wallabies. But what's really actually quite interesting in Victoria is that we've got a number of species that still persist in our more novel landscapes. So whether that's in our backyards, like the common brush-tailed possum, for example, you might even be lucky enough to have them living in the roof of your house, like I do. We've also got bush rats, for example, that do like to come into people's houses even in winter. And then we've also got the southern brown bandicoot, for example, that still persists on the outskirts of Melbourne, in those southeastern suburbs like Cranbourne, Bunyip and Cooey-Rup. And they've actually been able to persist in that sort of peri-urban agricultural matrix, even persisting in people's backyards and along drainage lines and roadside vegetation as well. Even in the broader northeast region where we are today, there are a number of microphagous mammals that still persist. So you're likely all familiar with many of these, but the yellow-footed antechinus, for example, still occurs in some of those drier forests in this region. We've got our brush-tailed phascogales, the tuan down the bottom, which still occurs in some of the forests, both in central Victoria and around Euroa itself. The yellow-bellied glider, unfortunately, is thought to be in quite low numbers or potentially regionally extinct in the Strathbogie Ranges itself, but they do still occur in relatively good numbers further eastwards and south of here. And then the long-nosed bandicoot, for example, inhabits some of those more denser vegetation types in this region. And then, of course, we've got some of these more common species again that you might be more familiar with, such as our wombats, bush rats, swamp wallabies, and possums again.
Cool. So a lot of these digging mammals, like our potoroos and bettongs, are termed ecosystem engineers because of the important roles that they play in maintaining ecosystem functions. So we know about their important role of eating fungi and dispersing their spores throughout the forest, but even by the very act of digging for these underground fungi, they're performing this process called biotubation, which is a movement of soil. And this has lots of different effects in the soil and the ecosystem in a more broader way. So this digging action in the soil creates more air in the soil and it increases the soil porosity, so more water can infiltrate into the soil. And these very digging pits themselves can act as traps for litter and for seeds. So it creates this nice little delicious environment for all these microbial decomposers to come in and start breaking down the litter and the seeds captured in this pit and increasing rates of nutrient cycling. Studies, there's been a couple that have also found that seeds that land in these pits have much greater rates of germination and growth when they're growing into a seedling compared to seedlings that are growing outside of these pits. So given that they're performing all these incredible roles in our ecosystems, what happens when they are threatened?
So many of our microphagous mammals were once common across much of Australia. However, as you can imagine, with increasing bushfire frequency and intensity, loss of habitat through conversion to agriculture or habitat fragmentation, and importantly, the introduction of feral cats and foxes across Australia has meant that many of these species have suffered reductions in population size, reductions in the range of their populations, or in some cases have gone either locally, regionally, or completely extinct. But obviously, when we lose these mammals from our landscapes, we risk losing many of the ecosystem functions that we've just described, relating both to the disturbance of soil and also the dispersal of fungal spores. And that's really thought to have contributed to declines in ecosystem health overall since European colonisation in Australia.
Oh, thanks. So this last slide of our introduction is really to drive home this three-way relationship between mammals, truffle-like fungi and plants, especially in the context of ecosystem functioning. So because we have so many mammals that disperse fungi, we don't really know what kind of functional redundancy we have here in the system. But we do know that having a diversity of mammals really increases ecosystem resilience. And we also know that all these different mammals are dispersing different kinds of fungi at different scales across different habitats. So we know that they're all playing their own really important role in helping get these fungal spores into contact with their host plants. And then onto the plants. So a lot of the dominant trees that we have in many of our forests in Australia are eucalypt trees. And they're ones that are predominantly forming a lot of ectomycorrhizal associations. So this kind of fills in this last link in the three-way relationship between mammals, tropholyte fungi, and woody plants. Alrighty.
Hand over to
Yeah,
you now.
thanks. So now I'm going to go into a bit of an overview of one of the projects that I'll be looking at for my PhD. I've just started my PhD at the start of this year, so I'm still narrowing down exactly what sort of experiments I'm going to perform. But this one is one that I'm really interested in carrying out, and it's to do with looking at the role of Potoroos in dispersing fungal spores after fire. So first I'll go into a bit of an overview of the two main species, or the two species of Potoroos that we have here in Victoria. So I've got the long-nosed Potoroo and the long-footed Potoroo. Potoroos, if you haven't come across them before, they're medium-sized marsupials that typically are found in quite dense vegetation. Our long-nosed Potoroo is mostly found along the coast of Victoria, so this is in the blue dots there on the map. And they're often found in vegetation types that might include coastal woodlands and heathlands, but also in dry eucalypt forests. Our long-footed Potoroo, on the other hand, is quite restricted in its distribution and is only present in two populations. So they've got one population up near the Barry Mountains in the Alps and one in East Gippsland. It's typically found in higher elevation forests. So these usually include dry eucalypt forests, wet eucalypt forests, and also in Gippsland, part of their habitat includes some patches of rainforest vegetation. So these mammals do spend a lot of time in really bushy, dense understory, which helps protect them from predators like foxes. And foxes in particular have been really damaging to Potoroo populations. But luckily, recent fox control efforts have been quite helpful in helping to protect these Potoroo populations. However, the long-nosed Potoroo is listed as vulnerable and the long-footed Potoroo is listed as endangered. So we still have a long way to go in helping to protect these animals. So we've discussed how important the special digestive system is in these Potoroos. It allows them to consume a really high diversity and abundance of different kinds of truffle-like fungi. There have been a few different studies that have looked at the diet of the Potoroo, both in assessing what kinds of and how many fungi that they're consuming. And this is typically done by having a look at their scats under a microscope and having a look at what spores are present. These fungal spores can then be used to kind of make identifications of what sort of fungi they're likely to be eating. So these studies have found that cumulatively, both these Potoroos are consuming upwards of 60 different species of fungi. The long-nosed Potoroo consumes fungi around 40 to 60% of its diet, whereas the long-footed Potoroo is our super fungal specialist, and it consumes fungi at over 80% of its diet all year round. So we know Potoroos consuming fungi, moving through the forest and pooping out their scat filled with all these fungal spores. But there's a couple of concepts that we should have a think about when we're talking about the scale of Potoroo dispersal of fungal spores. And the first one is the home range. So what is the potential range that Potoroos could be dispersing spores? There's been a couple of studies that have tried to estimate Potoroo home range size, and the estimates vary quite a bit. It's usually between 14 to 100 hectares. And this is quite a large home range compared to the size of these mammals, remembering that they're only about 30 centimetres in length. And it's thought that they need such a large home range because their diet, which is predominantly fungi, is spread quite sparsely through the forest. The second thing that we can have a think about is the dispersal distance. So how fast the Potoroos are moving compared to how fast their digestive system is moving. Estimates of Potoroo speed, there's been a few, and they've been found to travel multiple hundreds of metres in overnight setting. These animals are nocturnal, so they do a lot of their foraging and moving around at night. Then in terms of the gut retention time, or how long their gut is holding on to these fungal spores for, it's been estimated to be around 30 hours. So if we add these two measurements together, we could estimate that Potoroos are very likely to be dispersing fungi over multiple hundreds of metres, which is quite a lot when you consider that these fungi aren't going anywhere by themselves. So now on to our Potoroo populations in East Gippsland. The 2019 20 fires are outlined in that orange colour on the map there. And as you can see, they've burnt through 79% of the long-footed Potoroo habitat. So those are those red dots there. Remembering that these long-footed Potoroos are only present in these 2 locations in Victoria. Luckily, the Potoroos seem to be doing okay post-fire. They're still present at a lot of sites. They were present before the fire, but it's possible that with the fire destroying a lot of this dense vegetation understory that they need for protection, we might see increased rates of predation by animals like foxes. So if Potoroo habitat has been burnt so extensively by fire, what are they eating? So fire can have really varied effects on fungi. It can kill a lot of fungi in the top layers of soil, while a lot of fungi might be stimulated to produce a sporing body after fire. But some kinds of fungi, such as Mesophilia genus species, seem to be fire adapted potentially. They produce their fruiting bodies a little bit lower in the soil, up to 40 centimetres deep. So it's thought that they could be protected from the worst of the heat of the fire. And some studies have found that these are potoroid mammals, so mostly looking at the bettongs in these studies, have found that they start to consume Mesophilia in abundance after fire as a really high proportion of their diet. And normally these species are consuming quite a diversity of fungi. It's anecdotal evidence has found that these fungi, after a fire, they start to change their smell. And this is something that Alan just discussed. They start to smell like really stinky onions, which are a massive attractant to our mycophagous mammals. They just can't get enough of it. In Tasmania, looking at the eastern bettong, after a fire, researchers have found that they start to dig on these burnt sites really only up to days after a fire has gone through. So immediately after the fire, they're smelling these really stinky fungi and they're just going for it on these burnt sites. But they're also spending some of their time on the unburnt sites because they need that unburnt bush to have their nests and to get in that protective vegetation cover. So these animals are moving between burnt and unburnt patches of bushland. Now onto the Potoroos. So these populations of Potoroos in East Gippsland, they're likely to be interacting with a mosaic of burnt and unburnt habitat. And we, just on the other slide, discussed that it's probably likely that Potoroos are dispersing spores over multiple hundreds of metres. So are they playing a role in moving fungi between burnt and unburnt forest? And if they are introducing fungal spores back into burnt soil, is this having an associated effect on seedling growth? So this is the research that I'm hoping to be able to carry out as part of my PhD. So although the effects of fire can be quite variable, some studies in like the local area of southeastern Australia have found that hot fires can reduce the abundance of fungal fruiting bodies in the soil for up to a year after fire. Although some propagules might remain in the soil, so things like spores, where fungi can germinate and form more fruiting bodies, may still be present. Other studies that have looked at the effect of fire on ectomycorrhizal spores have found that fire decreases the diversity and abundance of spores in the soil. So we can hypothesize that on the unburnt landscape, we'll have this big diversity of fungal spores, whereas we might have a reduction in the number of spores on the burnt landscape. So given that it's likely that Potoroos are travelling between burnt and unburnt landscape, are they having an effect in moving the spores from the unburnt to the burnt land? So this experiment would involve collecting Potoroo scat and using it to inoculate seedlings growing on both the burnt soil, where there may be a reduction in fungal spores, and on the on the unburnt soil, where it's probably going to still be rich in spores. So my hypothesis for this question is that addition of fungal spores from the Potoroos scat, which will have this great diversity of fungal spores, will be able to introduce more spores into the seedlings growing on the burnt soil. And this can be assessed by looking at the seedlings after they've started to grow and having a look at which mycorrhizal fungi are forming associations with the seedling. Whereas on the unburnt soil, because it's likely that there's already a lot of different spores present, we might not see that Potoroos scat can add that many different kinds of spores. Now onto the second question, which is there an influence on seedling growth? Well, there have been some studies in Australia that have looked at inoculating seedlings with Potoroos scat, and these ones have found that if seedlings growing on sterile soil are inoculated with Potoroos scat, there's actually a big increase in the seedling growth. So this is great because it tells us that, one, a lot of these spores are still viable after going through the Potoroos digestive tract, and two, that they can support seedling growth compared to just growing on sterile soil with no kinds of spores. But we don't know what this looks like when their seedlings are growing in soil where there are all these different spores that might already be present. So I'm predicting that Potoroos scat, with all these fungal spores, adding more fungal spores onto the burnt soil may very well lead to an increase in the seedling growth. Whereas on the unburnt soil, already got lots of spores around, adding more spores might not necessarily increase seedling growth. So this is something that I'm really looking forward to carrying out and being able to see what the results of this are. So in terms of outcomes, for me this really fills in this third link in the three-way relationship between mammals, fungi and plants. So especially in the context of fire, because as we know, under climate change, we're probably going to see fires becoming more intense and frequent. So we know that Potoroos are dispersing these fungal spores around the forest, but are they able to introduce spores back into burnt soil? And if they are, what kind of effect is this having on seedling growth and potentially on helping seedlings regenerate after fire? So I'm really hoping this will give us a better insight into these endangered marsupials and their important fungal partners in post-fire ecosystems.
Thanks, Emily. So on to my project. So I'm about a year into my PhD at Deakin, and broadly my PhD is looking at what role can mycophagous mammals play in restoring ecosystem function. And today I'm just going to give you a bit of an overview of one component of my project that's looking at the fungal diet of a translocated mammal in Victoria. So we already highlighted that there's this really important role between plants, mammals and fungi, both through the dispersal of fungal spores by the mammals and then also that soil disturbance that can improve soil health. So obviously when we lose these mammals from our landscapes, we risk losing many of these ecosystem functions relating to both spore dispersal and soil disturbance. However, in recent decades, many of the conservation efforts across Australia have really looked at trying to reinstate these mammals into our landscapes. So whether that's trying to reintroduce a species that's been locally extinct, or maybe introduce a species outside of their historic range into some safe havens, for example. Many of the examples where this has occurred have really focused on reintroducing those mammal assemblages that we used to have in Australia and protecting some of these species from extinction. And so one of the methods in which we can reinstate these mammals is translocation. So it's simply the movement of an organism from one site for release in another. Sometimes this is from a captive bred population, so where we've taken them into captivity, such as into a zoo, bred up the numbers, and then we can re-release them. And sometimes this is just simply from one wild population to another. And some really familiar examples of this are the bilby, for example, in some of our arid landscapes, or even the eastern barred bandicoot over on the right there. And these two species in particular are very well known for their digging actions and have been found to improve soil health within our ecosystems, whether that's through water infiltration, increasing nutrient turnover, or providing some of those germination niches that Emily mentioned in our earlier introduction. And particularly within some of those arid landscapes where the bilby occupies, for example, their burrows can even provide habitat for other invertebrates and mammals, ameliorating some of those harsh climatic conditions that you get in those arid landscapes. There's also been lots of examples of mycophagous mammals that have been translocated across Australia, like our bettongs or our potoroos or our bandicoots, for example. And obviously in doing so, we reinstate those processes relating to fungal dispersal and hopefully therefore have beneficial responses in plant growth. So I just thought I'd go into a couple of well-known examples. For example, the burrowing bettong has been reintroduced into a fenced enclosure called Arid Recovery over in South Australia. And so fenced enclosures are really, really common in Australia because essentially they remove those threats from predation from feral cats and foxes. And in many cases, this is the only way that we can currently reintroduce some of these species. Otherwise, the threat of predation is just too high, they would simply go extinct again. And similarly with the eastern barred bandicoot down in Victoria here, they've been reintroduced into fence reserves in Victoria, including out at Hamilton in the west or even up at Woodlands Historic Park near the airport. In other cases, offshore islands have provided safe havens where the water itself provides a fence of sorts to prevent feral predators from entering. So for example, the Gilbert's Potoroo in WA, or even again with the eastern barred bandicoot who have now been introduced onto Phillip Island. And then in other cases, for example, up in New South Wales at Booderee National Park, following really extensive and long-term fox control, they were able to successfully reintroduce the long-nosed Potoroo and the southern brown bandicoot. And I suppose a really key theme with all of these examples is just that we need to really remove those threats from predation to be able to successfully reintroduce many of these species. So this component of my PhD is aiming to understand what role a translocated mycophagous mammal can play in restoration projects. And in this case, we're looking at the eastern barred bandicoot. So the eastern barred bandicoot is a generalist mycophagist. So they feed on fungi seasonally and use it to supplement their diet rather than relying on it for the bulk of their diet. And so along with fungi, they feed on a range of invertebrates, such as your worms, your beetles, as well as seeds, plant roots, onion weeds even. And one previous study on the eastern barred bandicoot down at Churchill Island and Phillip Island found that bandicoots fed on fungi primarily in winter and spring, but there hasn't actually been a study looking at the specific fungal species or taxa that they feed on. So we don't really know what role they're having in terms of fungal dispersal. You know, are they dispersing more of those truffle-like fungi? Are they incidentally dispersing more of those arbuscular-like fungi that associate with more of those grasses while they're digging in those grasslands in search of other food sources, such as invertebrates? So eastern barred bandicoots were once widely distributed across the western volcanic plains of Victoria. However, following the introduction of feral cats and foxes and the conversion of much of these landscapes to agriculture, they actually went extinct in the wild in the late 80s. And just before they went extinct in the wild, a captive breeding population was set up, and that population was established just from a single population that was out at Hamilton. So essentially our current bandicoots come from just one population in Victoria, nowadays with a few extra genes thrown in from the Tasmanian population of bandicoots as well. So following their successful breeding program at Zoos Victoria, they then reintroduced them onto a number of fenced reserves, which includes Woodlands Historic Park and Hamilton Community Parklands, which are the two here shown by the orange arrows. And I'm using these two sites, as examples of their historic range for my study. Then, unfortunately, in the past sort of 30 or so years, following variable success in the introduction of the eastern barred bandicoots into these fenced reserves, due to a combination of either fox incursions, we had the millennium drought, there was overgrazing by macropods, which then removed the grass cover for the bandicoots and then removed food resources, and also the inability of some of these smaller sites to really maintain a genetically diverse population of bandicoots. They were then introduced outside of their historic range onto 3 offshore islands off the coast of Victoria. So Phillip Island, Churchill Island and French Island, just shown in the bottom here by the blue arrows. And I'm using these 3 sites as examples of an introduced population of bandicoots, so outside of their historic range. So basically I'm going to be using scat analyses to look at the bandicoot's fungal diet. And to collect the scats, we've been joining the annual population monitoring that occurs at each of these sites. And this involves the use of cage traps. Some of you might be familiar with them. They're non-lethal traps that are very commonly used in zoology to monitor animal populations. In this case, we bait them with a delicious combination of peanut butter, golden syrup and oats, much like an Anzac cookie. We then leave them out overnight or over the course of several hours, and then we go back and hope that there's a bandicoot in them. This year we had really good numbers actually, so often we did have a bandicoot in them, which was quite a success compared to some of the earlier trapping sessions that they did. And when we get to the bandicoot, we also undertake a number of measurements to keep track of their population. So we sex the animal, we see if they've got pouch young, we assess their body condition, we weigh them, and we also put a microchip on them just to keep track of those populations so we can track individuals over time and see how the population's growing, hopefully. And in this case, I've been collecting scats from the base of the traps that the bandicoots hopefully have left behind for us, but isn't always the case. Perhaps we need to feed them more peanut butter. So after I've then collected all the scats, I'm then going to be using DNA metabarcoding to identify the fungal OTUs, or the fungal species within them. So yeah, that will be interesting because nobody's really looked at the specific species that the bandicoots feed on. So we're really just trying to look at here whether there are differences in their diet, either between sites, which might be related to whether they're inside or outside their historic range. It could also be related to the type of vegetation. So some of the sites are more grassy, for example, whereas others have more established woodlands. So potentially in the woodlands sites, we're expecting more ectomycorrhizal to be present. And then another component I'm also going to be looking at is the comparison of the fungal diets of the eastern barred bandicoots with 2 species that were already present on 2 of the islands, so Phillip Island and French Island. So down at Phillip Island, there's an established indigenous population of swamp wallabies, and similarly to the eastern barred bandicoots, the swamp wallabies are a fungal generalist, which again means that they feed on fungi seasonally when it's most abundant. And over on French Island, there was an established population of long-nosed Potoroos, which are fungal specialists. And the comparison with these 2 other species enables us to look at differences in diet between these generalist and specialist species in the case of French Island, but it also enables us to compare the diet of both that introduced eastern barred bandicoot with that of existing mycophagous mammals on each of these islands. And we're looking at this to really understand what niche the bandicoots might be fitting into within these existing landscapes, whether they might be value adding to services related to those ecosystem functions that we've mentioned, and sort of how they're interacting in their new recipient ecosystems. So hopefully this component of my project will build our understanding of the fungal diets of the eastern barred bandicoots, and in doing so can give us an indication of what influence they might be having on the fungal community dynamics and by extension the plant community dynamics. Hopefully we can also contribute to a growing body of research on translocations that's been looking at how translocated mammals impact the ecosystems that they've been introduced into, and also potentially feed into future translocation programs either of the eastern barred bandicoot or of other mycophagous mammals onto these two islands. And lastly, I just thought I'd encapsulate my entire PhD project through this figure here. So really we're honing in on those mycophagous digging mammals and trying to understand how they fit into the broader landscape of our ecosystems. So how they interact with other species in their community, whether that's our fungi, our plants, or other mycophagous mammals. What functions they then perform within our landscapes, whether that's related to soil health through digging or whether it's related to fungal spore dispersal, and therefore how we could then apply these species within a restoration context for the provision and restoration of some of these ecological functions. And I just want to thank these organisations for providing financial and logistical support thus far. And before I finish, I also just wanted to plug another component of my PhD that's actually looking at the relationship between digging mammals and soil health, both in the context of agricultural landscapes and in reserve systems as well. And part of this component's going to involve a questionnaire or a survey type questionnaire that will be sent out to farmers, landholders and conservation organisations just to gauge people's perceptions of digging mammals in these landscapes, what soil health properties you find are important, and current management practices, whether that's on farms or in reserve landscapes, and how we might be able to integrate digging mammals into these conservation plans. So if you're interested in being involved, please let me know. The survey will come out later in the year, and otherwise just keep an eye out on some of your local environmental channels, because we'll be releasing it through local land cares, catchment management authorities and alike. Thanks very much. Oh.
Wow, Emily,
Yeah.
I forgot.
they sound like awesome projects. And it's great that there's actually a couple of people sort of doing similar tracks so that you can actually support each other.
Yeah.
I've actually got several questions, but just in case there aren't too many, but maybe there are some. Any questions, or do I have the floor? Sue, thanks.
Oh, my question's a bit off to the side here.
Oh, no, another one then.
It's quick. I noticed in one of the slides with your truffle-like fungi that it looked like what I call horse-dung fungi that was sort of sticking up out of the ground a bit. But it might not have been that one. But what I'm curious about, I noticed, is how people have found these things, like because they're under the ground, like unless you inadvertently, like I've had friends come up and ask me, what is this, they've been digging something, they've found it, but how ecologists have found these species here.
Oh, I think it's just through strategic and slow surveys. Yeah, probably, it's very disruptive, but you tend to go out from a tree, for example, so you've got the root system and then you basically work your way out and look for truffles, but it's quite disruptive. Yeah, but I don't know if...
I've read some studies they use raking, like they'll choose a plot, or if they want to just do a base around the base of a tree, and they'll use a rake with a certain, centimetre tines on the rake and kind of dig through the dirt and sift through to find the different sporine bodies. But yeah.
Unless we can train Potoroos to sniff out the truffles for us, which would be great.
It's also fair to say that it's fairly recent. This line of research is fairly recent.
Yeah.
And it really only follows on from, the detailed work in those Pacific Northwest forests on, squirrels and so those northern squirrels and the old growth forests, that some of those, and I saw trap mentioned in somebody.
Yes.
When they came over and started looking at similar systems here. So we're only looking at maybe 40 years of understanding of that whole mycophagous process here in Australia.
Yeah.
And you were going to, sorry, I interrupted you.
There's so many things that I want to say. I was just going to say to what you said then, like the long-footed Potoroo was only described formally in 1980, so research into it is still pretty new as well.
But still, 1,500 of those are underground fungi.
Yeah. So
someone's documented.
Yeah.
So that number's actually an estimated number. So I don't think that many have been described, but I think based on how many are described and which locations and over how much time, that's like the estimated total number in Australia. But some of the authors that have done these papers raking through the soil have remarked that like the mycophagous mammals that they're studying find so many more different kinds of fungi than they can find in their like systematic searches. So yeah, I think Aviya was on the money about training Potoroos to find fungi for us.
Thank you.
Sally.
I hope this is not a mean question.
I know.
I'm always amazed, by experiments conducted in the wild, because there are so many factors to control, and it's probably really difficult to remain clear-headed about what you're looking at. But one thing that occurred to me was in the burnt, unburnt seedling growth observations, how do you discount the fact that in the burnt area, seedlings might grow faster because there are more immediately available nutrients and less competition from other plants? How do you sort of separate that out?
Yeah, that's a really good question. I think for my experiments, I'm planning on doing a greenhouse experiment. So be taking soil from these places and growing it in a greenhouse. And something that I'd like to do in addition to just looking at soil from burnt area and non-burnt is also getting soil from burnt area and experimentally burning it. So taking a core and then taking it into the lab and applying different degrees of heat to it, which will then, it's not the same thing as fire, but that might be one way that you can kind of remove that factor of this big addition of nutrients from the burning of all the canopy species or whatever it is. So obviously I can't do every single test that I'd like to do, but if I could do those two things and compare them, I feel like that would be really helpful in trying to break apart what might be some of the influencing factors.
One of the things being repeated all the way through our presentations is the relationship between fungi and trees. Now, I'm guessing it's really fungi and plants, is it not? Grasses, shrubs, everything?
Yeah, definitely. Like a lot of plant species form these mycorrhizal associations. I guess
it's just that the truffle-like fungi, for example, that we were sort of focusing on, associate mainly with trees and woody shrubs, compared to your more like arbuscular mycorrhiza that associate with those herbaceous and grassy vegetation. And I guess in most cases, the truffles rely more on the mammals rather than the arbuscular to disperse their spores, that is. Yeah.
So you mentioned a couple of times that the stomachs of the mycophagous mammals are specialised. Is that in anatomy? Is it in chemistry? Is it because chitin is notoriously hard to break down?
Yes.
So is it related to that?
Yeah, so it is related to the physical anatomy. So they have, I can't visualize it in my mind, but they have an enlarged foregut, which is like a part of their digestive system. And this allows them to use microbial fermentation in this part of their digestive system. So I guess all these microbes are helping to break down the fungi. Yeah.
I was just wondering if when your research goes down the track a bit more, you find out exactly what funguses that they're eating. If you'll be able to find out if they're the truffle-like funguses that toxic or not toxic to other animals, such as humans. Yes, I'm interested to know if they're able to eat fungi which would be toxic to anyone else, but they can eat.
Was this to one of us in particular? Or?
Yeah.
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 Are you passing the buck?
Yeah.
I can go for it.
Yeah, you can go for it. Yeah.
Oh, I was going to say, probably a lot of these underground fungi, we don't know, if they have toxins or what kind of toxins they have. I know some fungi have got particular genes that we know this gene produces this toxin, but I think it's likely for a lot of our Australian species we just don't know. Yeah, that would be my answer to that.
Yeah.
I don't know if you have anything extra to add.
That sounds about right. Yeah.
I like very much to avoid work. Digging doesn't sound very attractive to me. And as an engineer, of course, you regard that as efficient. But it occurs to me if you really want to look at roots of trees, especially larger trees, the most efficient thing to do is to wait for a windstorm and wait till they dig themselves up. Do truffles grow on any particular size of tree? The big trees do get blown over, but do you find that truffles occur on bigger or smaller trees?
I'm actually not sure myself.
Often some of the eucalypts will start off with more of a dominance of arbuscular, more so when they're little, and then they'll get more truffles later on. But I mean, correct me if I'm wrong, Camille, but yeah, they sort of, I think they'll still be on smaller individuals, but yeah.
There are some species that are more specific to seedlings, and then by the time the tree gets bigger, its demand in nutrient is also much bigger, and so it kind of turns to other species. So this kind of, if you watch from like a seedling stage to another tree, you have some kind of succession of different species along the life stage.
And just to satisfy my curiosity, yellow-bellied gliders,
Yes.
if I remember correctly, they don't spend a lot of time on the ground.
Yes.
So are they eating bracket fungi or what? Or
I'm not sure what what fungi specifically they're feeding on, but I can come back to you with that. Would anyone else know?
Incised trees and
Yeah.
drink the sap. I imagine they encounter lots of fungi on the way up and down the tree trunk as well. But yeah, whether they're those sorts of fungi that you guys are talking about.
Yeah, those lists were just general fungal feeders. Yeah, I can't imagine that they'd be digging on the ground, but from an
they might be eating bracket.
No, definitely, yep.
Well, let's just give Aviya and Emily a round of applause.